Hello, welcome to Biostudy Pathshala, and I'm Nilanjana Sharka, wishing you a very good morning and a very good day ahead. Today's topic is Auxin phytohormone, and this is part 2 session of the Auxin phytohormone and the continuation of the previously uploaded video of the Auxin phytohormone. So let's start today's session. We will learn about Oxyinduced cell extension procedure. So if we want to simplify this term, that is cell extension or the cell enlargement procedure by Auxin induction. This particular event is also known as acid growth hypothesis. This hypothesis states that H+ ions, that is hydrogen ions, may act as the intermediate molecule between Auxin phytohormone and the cell wall loosening factor, that is expansin. This H+ ions provides acidic environment to the Auxin phytohormone, which is optimum for the Auxin phytohormone. The source of the H+ ions are H+ ATPase channels, which are situated on the plasma membrane. This H+ ATPase channels activity may increase the response of the Auxin phytohormone. Auxin phytohormone needs an acidic pH to act in its optimum level. It cannot act in neutral pH because neutral pH may inhibit the Auxin-induced growth, and this Auxin phytohormone results in formation of the cell wall loosening factor named expansin, which ultimately results in the cell enlargement procedure. We will learn this topic in detail, so stay tuned till the last of this video. Auxin-induced proton extrusion may occur by two possible mechanisms. One is activation of the pre-existing H+ ATPase channels in the plasma membrane, and second one is synthesis of new H+ ATPase channels on the plasma membrane. That means first one states about the pre-existing H+ ATPase channels and activation of those channels, and second one states about synthesis of new H+ ATPase channels on plasma membrane. Just have a look on this figure. We will learn these two possible mechanisms one by one corresponding to this particular figure. Here ABP1 stands for auxin binding protein. This auxin binding protein binds to auxin molecule, that is IAA, and forms ABP1 IAA complex. Okay, and this particular is H+ ATPase gene, and this is the H+ ATPase mRNA. So now we can start these two possible mechanisms one by one. Okay, first one is activation hypothesis. That means the activations of the pre-existing H+ ATPase channels. In this figure, these are the H+ ATPase channels. Okay, and according to this hypothesis, auxin binds to ABP1, that is auxin binding protein, and forms ABP1-IAA complex, and this ABP1-IAA complex activates these H+ ATPase channels. The activation of this particular H+ ATPase channels stimulates the proton pumping. That is why the H+ ions which were in cytoplasm area extruded to the cell wall portion. Sometimes here calcium as well as intracellular pH acts as secondary messenger, which can proceed the second hypothesis, that is the synthesis hypothesis of H+ ATPase channels. We will learn now the second hypothesis. So second hypothesis is synthesis hypothesis, that is the H+ ATPase channel synthesis procedure. Here IA molecule diffuse directly into the cytoplasm and produces secondary messengers, that is intracellular pH or calcium ions increasement. These intracellular pH and the calcium ions induce the transcription of this particular gene, that is H+ ATPase gene, and forms H+ ATPase mRNA. This H+ ATPase mRNA then goes to rough endoplasmic reticulum to translate H+ ATPase protein, that is H+ ATPase channels, and these H+ ATPase proteins after synthesized goes for the plasma membrane through Golgi bodies, secretory pathway, and this is how H plus ATPase plasma membranes are synthesized, and this H plus ATPase plasma membrane ready to pumps proton from cytoplasm to cell wall, and the accumulation of the H plus ions in the cell wall portion may activate the expansin protein, that is the cell wall loosening factor, and the accumulation of the expansin protein may result the enlargement of the cell wall, or the cell wall loosens by this expansin protein. Okay. Next one is auxin induced gene. Auxin induced genes are classified into two. These are early genes and second one is late genes. Early genes are those genes which are activated by the pre-existing transcription factors, and late genes are those genes which are activated by the early genes. Early genes further divided into two families of genes. These are growth and development related genes, and second one is stress related genes. Growth and development related genes again classified into three categories. These are Auxin gene family, SORD gene family, and GH3 gene family. Okay, Auxin gene family may act as either repressor or activator in auxin signal transduction pathway. We will learn about the auxin signal transduction pathway in few minutes. Okay, the product of this particular gene family are short-lived and may act as either repressor or activator. SORD gene family may express within 2 to 5 minutes after the exposure of the auxin phytohormone, and its response is insensitive against cyclohexamide. Okay, next one is GH3 gene family. The product of the GH3 gene family again produced within 5 minutes after the exposure of the auxin phytohormone, and this GH3 gene results in high growth of Arabidopsis plant because the mutant of the GH3 gene product shows dwarfism in Arabidopsis plant. This particular gene family identified in soybean plant as well as Arabidopsis plant. Okay. Second one is stress related genes. That means these genes are related to stress environment in plant body. These are one glutathione transferase gene encoding family, and second one is SEC synthase gene encoding family. Glutathione ACE transferase, that is GST, stimulated in various stress conditions, and these genes are induced by auxin in particular stress conditions. The full form of SEC is 1-amino cyclopropane 1-carboxylic acid synthase. Okay, this particular gene is the key enzyme in case of ethylene biosynthesis pathway. That means this gene product may lead to the production of ethylene phytohormone, and this ethylene phytohormone is synthesized in case of stress time in plant body. Okay, so next one is auxin signal transduction pathway. Before starting about this signal transduction pathway, we have to know some terminologies. We have already known about Auxin family, that is repressor or activator family. Here it acts as repressor protein. The full form of ARF is auxin response factors, and second one is AuxRE. AuxRE stands for auxin response elements. Okay, in this case auxin, that is IAA, is absent, and in this case auxin, that is IAA, is present. Now you will see the consequences when auxin is present and auxin is absent. Here as you can see, auxin is absent. That is why auxin cannot bind to this particular repressor, that is AuxIAA protein. This results to formation AuxIAA ARF heterodimer, and this particular heterodimer inactivates the transcription procedure of AuxRE gene. That is why here transcription is off. But in this case, IAA is available to bind to AuxIAA protein. After binding, IA-AuxIAA complex dissociates the ARF protein. That is why ARF is free to form the ARF homodimer, and ARF homodimer formation is very crucial to make the transcription procedure on, and the formation of the ARF homodimer leads to transcription on. That is why transcription procedure here proceeds. And this particular complex, that is IAA Ox-IA complex, goes for 26S proteasomal degradation by the machinery of the 26S proteasome and degraded it. Okay. So that is how auxin signal transduction pathway occurs. We have already discussed some of the functions of auxin in our previously uploaded video of auxin. You can watch from particular that video. Now we will discuss rest of the functions. First one is delay in abscission. First we have to know the abscission. Abscission means shedding of plant parts. It might be flower, fruits or leaves. As you can see in this figure, these are the abscission zone. Abscission occurs from the abscission zone or abscission layer. Auxin prevents this particular abscission procedure. Particularly in premature leaves or premature fruits and flowers, auxin prevents the abscission, and during leaf senescence, when the abscission triggers, auxin is no longer being produced in those particular parts. That is why abscission occurs. Next one is Oxyinduced vascular differentiation. Auxin may induce the vascular differentiation in case of tissue culture when apical bud is placed on callus. The callus results in formation of xylem and phloem beneath the graft. When concentration of auxin is so much high, then it leads to formation of xylem and phloem. But when the concentration of the auxin is low, then it leads to only phloem differentiation. That means to differentiate xylem and phloem both, we need high amount or high concentration of auxins, but to differentiate phloem tissue, we require only low concentration of auxin. Okay. And after wound in plant body, the regeneration procedure of the vascular tissue also require the auxin phytohormone, and this auxin phytohormone then produced by the young leaf directly above the wound site. As you can see in this diagram, the leaf is present and here a wound you can see, and the vascular differentiation takes place above the wound region. Okay. And the vascular differentiation procedure is a polar incident which occurs from apex to base, that means from leaf to root. Next one is auxin regulations in floral bud development. Floral bud development requires pin protein and as well as auxin phytohormone. How we could know about this particular requirements? The NPA treated plant, that is naphthalamic acid treated plant, mainly Arabidopsis, shows the abnormal floral development. NPA can inhibit the auxin phytohormone effects. That is why the NPA treated Arabidopsis plants show the abnormal floral development, and the pin protein is required for the polar transportation of auxin. We have already learned about the polar transportation of auxin in our previous auxin phytohormone session. So if the pin protein is mutated, that means pin protein is not able to transport auxin in polar way. That is why the mutant pin protein shows also abnormal floral development. You can see this is normal floral development with adequate amount of auxin, but this figure shows abnormal floral development. Either it is treated with NPA or it is with mutant pin protein. Okay. Next one is auxin in fruit development. Auxin can induce the parthenocarpic fruit. Parthenocarpic fruit means the seedless fruit production. So the application of the auxin phytohormone may induce the parthenocarpic fruit production. It also prevent the premature abscission, that is fruit and leaf drop. It can promote the pineapple flowering, as well as auxin may induce in plant propagation. Auxin can observed in pollen as well as an endosperm embryo of the developing seeds. The developing seeds require auxin for initial stage of growth, and endosperm contribute the auxin during the initial stage of growth. Okay. Next one is auxin as herbicides. 2,4-D, that is 2,4-dichlorophenoxyacetic acid, and dicamba are well known synthetic auxins which are used as herbicides. Why synthetic auxins used as herbicides? Because synthetic auxins are not so easily metabolized in plants as natural auxin, that is IAA. So it is very effective herbicides. And last one is ethylene biosynthesis. We have already known that the stress related genes, which is auxin induced, that is ACC synthase, may be induced by auxin in case of stress time, and auxin may play role in ethylene biosynthesis by the induction of the ACC synthase gene, and this ethylene is involved in fruit ripening, and this is the biosynthesis procedure of the ethylene biosynthesis. Although we will learn about the ethylene biosynthesis procedure in our upcoming plant physiology class, so stay tuned with Biostudy Pathshala. This is for today's session, guys. Thank you for watching this video, and if this video is helpful for you, you can give feedback on comment section below, or you can like this video. And lastly, don't forget to share this video and subscribe this channel, and if you want to get notifications from this channel whenever new video is available on this channel, you may press the bell icon below. So till then, be safe and be healthy. Thank you. Bye bye.